Hey everyone, and welcome. Today, we're going on a pretty incredible journey. We're going to go from the absolute smallest bricks of life all the way up to the grand blueprint that makes you, well, you. So let's dive right in. So here's the plan. We're going to start with the basics, what I'm calling the bricks of life. Then we'll clear up a super common point of confusion, nucleosides versus nucleotides. From there, we'll zoom out to the master blueprints, DNA and RNA, figure out how to decode their language, see how all that information actually turns into life, and then, finally, we'll bring it all back home to you.
All right, let's kick things off with section 1, the bricks of life. I want you to imagine you're building something incredibly complex, like a massive Lego castle. You have to start with that one single fundamental brick. Well, in biology, that brick is the nucleotide.
So that's the big question, right? What are these fundamental little pieces that make up your DNA and really all of life as we know it? And the answer is the nucleotide. You can think of it as that single repeating brick. Scientists have a word for this, they call it a monomer, and it's what you chain together over and over again to build the giant structures of DNA and RNA. It is the absolute foundation.
And each one of these little bricks has 3 distinct parts. It's actually pretty simple. You've got a nitrogenous base. That's the unique letter of the code, A, T, C, or G. That letter is attached to a sugar, which forms the sturdy backbone of the chain. And then finally, you have a phosphate group, which acts like a connector, linking one brick to the very next one.
Okay, this is where it gets really interesting, because nucleotides aren't just for building DNA. No way. They are the ultimate multitaskers inside your cells. Yeah, they carry genetic info. That's their main job. But they also act as the energy currency for the entire cell. You've probably heard of ATP, right? The molecule that powers basically everything you do. That's a nucleotide. It's amazing.
Okay, next up, let's clear up a common little stumbling block, the difference between a nucleoside and a nucleotide. The names are so similar, it's easy to get them mixed up, but the distinction is actually super important. And it all comes down to one tiny little part. This table just lays it out perfectly. A nucleoside is just the base plus the sugar. That's it. But the second you add that phosphate group, bam, it becomes a nucleotide. That single addition changes everything. The phosphate is what gives the nucleotide all its power. It's the connector that lets it build long chains. And it's the battery pack that lets it store and transfer energy. It's the key.
Okay, so we have our fundamental bricks, the nucleotides. Now let's step back and look at the incredible structures they built. We're moving on to the master blueprints for all of life, DNA and RNA.
So these long chains of nucleotides, they have a formal name, nucleic acids. Think of it like a train made of many, many repeating cars. A nucleic acid is a polynucleotide, which is just a fancy way of saying many nucleotides all strung together. And their whole job? To store and pass on our genetic information. And first up, we have the celebrity of the group, DNA, or deoxyribonucleic acid. This is the master blueprint. It's that beautiful, iconic double helix structure. It's incredibly stable, and it's designed for one thing, the long-term, super secure storage of all the instructions needed to build and run an organism. Now, if DNA is the master blueprint locked away safely in the architect's office, then RNA, or ribonucleic acid, is the working copy you take out to the construction site. It's usually just a single strand, and it's designed to be a short-term messenger. Its job is to carry one specific instruction from the DNA out to the cell's machinery that's actually going to build things.
And this table breaks down those key differences so nicely. DNA is double-stranded, RNA is single. They use slightly different sugars in their backbone. But look at the bases. DNA uses thymine, or T. RNA swaps that out for a very similar base called uracil, or U. It's almost like a little watermark that tells the cell, hey, this is just a temporary RNA copy, not the original blueprint. Okay, so we've got these long strings of letters. But how does the cell actually read them? I mean, how does it make any sense of it? This brings us to decoding the genetic language, turning all those individual letters into meaningful words. The genetic code is basically the dictionary. It's the official set of rules that tells the cell's machinery how to translate that sequence of nucleotide bases into a sequence of amino acids, which, are the building blocks of proteins. It's literally the instruction manual for life. And this language has some really fascinating properties. First, it's a triplet code, which means the cell reads the letters in groups of 3. These are called codons. It's also degenerate,
which is a weird word, but it just means they're synonyms. Different three-letter words can code for the exact same amino acid. And maybe the most incredible part, it's nearly universal. The same codons mean the same thing in you, in a tiny bacterium, or even in a giant redwood tree. Let's do a little bit of math here, because it reveals something so brilliant about the system. You have 4 possible letters, right? And the cell reads them in groups of 3. That means there are 64 possible 3-letter words, or codons. But, and here's the twist, there are only about 20 different amino acids used to build proteins. So what gives? It means we have a bunch of extra codons. This isn't a mistake, it's a feature. This built-in redundancy is like a safety net, protecting the cell from typos or mutations. All right, we've got the blueprint, we know the language to read it. Now for the big one. How does that static information on a page, so to speak, actually become the dynamic, living stuff of our cells? Well, this process is explained by something called the central dogma of molecular biology. It's this beautiful 3-step flow of information. First, you have replication. That's when DNA makes a perfect copy of itself, which is obviously crucial for when cells divide. Second, you have transcription. This is the process of creating that working RNA copy from a gene in the DNA. And finally, translation. This is where a ribosome grabs that RNA message, reads the codons, and translates it into a specific protein. It's a perfect system, from storage to message to action.
So let's zoom all the way back out. We've gone from the tiniest nucleotides up to nucleic acids and the genetic code. How does all of this connect back to us, to our traits, to our families? Let's put it all together.
The key concept here is the gene. A gene isn't just some vague idea, it's a physical thing. It is a specific segment of that long DNA molecule that contains the complete set of instructions, the entire recipe for building one specific protein. If you think of your DNA as a giant cookbook, then a gene is just a single recipe inside of it. And inheritance? Well, that's simply the process of passing that whole cookbook down. It's how all that genetic information, the entire collection of recipes, gets transmitted from one generation to the next. And that is the fundamental reason why families share traits and look alike. And this really just nails it, right? This quote from Dr. Ruxana Ribose in our source material sums it all up perfectly. Nucleic acids are responsible for the transmission of inherent characters from parent to offspring. Everything we've talked about, from the smallest brick to the grandest blueprint, it all comes back to this one fundamental job. So we'll end with this thought. From a single nucleotide brick, to the DNA blueprint, to the genes passed down through your family, it all works together to write the story of you. Which kind of begs the final question, how much of that story is already written in permanent ink? Thanks so much for joining us for this explainer.